Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjong classes. Dear students, today we are starting with the one of the new chapter, that is the cell, which is a unit of life. The unit of life, that is cell. Now, dear students, this chapter is a part of cell biology. Okay, and we are going to deal with the cell in this chapter in detail. We know that cell is a structural and functional, basic structural and functional unit of life. So if we talk about this, first the question which we are going to discuss here, that is what is the basic unit of life? Dear students, if you see any organism, either it is unicellular, made up of single cell, or it is made up of a multiple cell or multicellular organism, ultimately whatever the reactions which is responsible for the survival of this organism that occur within the cell. So cell is the basic unit of life because it carry out all the necessary functions which are responsible for the survival of the cell as an overall body. What is meant by biology? If we talk about biology, it is the study of form, function, and interrelationship of living organism. It is the study of form, function, and interrelationship of living organism. Now, if we talk about a cell structure, the first study, if we talk, that is initially done by Ramchandran, and he is an outstanding figure in the field of protein structure. He done a very good research in the protein structure. His discovery of the triple helical structure of a collagen was published in Nature in 1954. Okay, and this triple helical structure of this collagen protein given by Ramachandran, and from that, the sum of the discovery with related to cell was started. Now the question is, how many basic things are there when we look around us? We see both living and non-living things. There are several living organisms, from a microbe, from a small bacteria to a large higher plants and higher animals we can see. And we also see some of the non-living things. Now, if we talk about this living and non-living things in detail, we know that organisms are living. So the question is, what is that makes an organism living? So the presence of basic unit of life, the cell in all living organism, makes them living because the cell carry out all the necessary functions which are required for the existence of a living organism. And due to the cell, the property is given to the tissue, then tissue will give a property to the organ, and further it will give a property to the organ system, and such a way property ultimately is given to the organism. So this living nature, if we talk, that is due to the presence of basic unit of life, that is living organism, that is the cell. And we know that all organisms are composed of cell. And we know that there are two different types of organism mainly based on the number of cells. They are the unicellular and multicellular. Unicellular organism made up of single cells, and multicellular organism are made up of many cells. If we talk about unicellular, which are made up of single cell, there are so many examples in unicellular organism, like bacteria are there which are unicellular. Then we can say some single cell yeast are there, fungus single cell fungus, that is yeast is there. Then some protists are there, protozoans are there, and some algae, unicellular algae are also there. They are all are made up of single cell. But when we talk about some higher plants and higher organisms, higher animals, they all are composed of many cells. So by studying this much things, we are now clear that cell is the basic unit of life, and cell is the ultimate region for the survival of any living organism or the existence of any living organism. So now we have to understand what is cell. So if we talk about a cell, cell is the fundamental structural and functional unit of all living organism. It is the fundamental structural and functional unit of all living organism. Suppose it is a structural and functional unit of all living organism, then unicellular organism and multicellular organism, if we talk, these are unicellular organism, these are multicellular organism. Unicellular organism are capable of independent existence and performing the essential functions of the life. Means, if we talk unicellular organism, they are made up of just a single cell. A single cell has a capacity to carry out all the functions whichever required in for the survival of this single cell organism. So, due to the presence of single cell, on all activities performed by the single cell, they are independent. They show independent existence. Existence. They live independently, and with that, they perform some essential functions of life as well. Now, later on, if we talk about unicellular organisms are capable of independent existence and performing the essential functions of life, anything less than a complete structure of cell does not ensure independent living. Be remember, any cell, anything which is less than a complete structure of cell, which does not show a independent living. Hence, cell is fundamental, structural, and functional unit of organism. So now, in the discovery of this cell and the study of cell, one of the important name of the scientist is Antony von Leeuwenhoek. If you talk about Antony von Leeuwenhoek, he was the first to saw and describe a living cell, and he described the first living cell. And this is the some diagrammatic representation of microscope and the images which was developed. Now, what Antony van Leeuwenhoek did, that is the important thing. He first saw and described the live cell. He saw some RBCs, some protozoans, and see them they are capable of moving and they perform an important independent function of their life, and they call it as a live cell. But later on, if we talk, then within a cell, when he is seen that there is a presence of nucleus. So now the question is, who discovered the nucleus? So Robert Brown later discovered the nucleus, and we know that the invention of the microscope and the improvement leading to the electron microscope revealed all the structural detail of the cell. So electron microscopy revealed all this structure. So Robert Brown discovered the nucleus. We know that nucleus have a particular this type of structure. We will discuss this nuclear in the later part. And we know that nucleus is found in eukaryotic cell, but true nucleus is not found in case of the prokaryotic cell. That is the one of the important thing which is seen in case of the presence or absence of nucleus. Now, further, after this discovery, if we talk, cell theory came into the picture. Before that, we should remember this important discovery that who is the famous scientist in the field of protein structure. And we should know that it is nothing but G.N. Ramachandran. Triple helical structure of collagen was discovered by Professor P. Maheshwari, M.O.P. Iyengar, G.N. Ramachandran. Thus, we have studied that it is a G.N. Ramachandran. Structural and functional unit of organism. If we talk, we know that the basic unit is the structural and functional unit of organism, and that is nothing but a cell. Okay. Now, further, we discuss about the modern term of our cytology. Cytology is also a study of what a cell, but modern term for the cytology is nothing but a cell biology. Next question is, who introduced the term cell? We remember the term cell was introduced by Robert Hooke, and Robert Hooke was the first discovered cell. So he first cell discovered by Robert Hooke in Cork. Now the question is what he did. So that we should understand. Then we will come to the cell theory part. So let's discuss about some of the initial discoveries which was made for the discovery of cell. Okay, and if we talk about that, so we should understand the topic is discovery of cell. If we talk about discovery of cell, the work on the study of cell has continued far more than last 3.5 centuries. It required microscopes or instruments with good resolving power and magnification. There are some important techniques are also required for this magnification, and important improvement in tools and techniques has also continued all this period to enhance the our knowledge about the cell. Now it is known that the discovery of microscope is important for the study of cell, and it is known that first microscope was built by was built by Zacharias Janssen in 1590. So first microscope was built by Zacharias Janssen in 1590, and it was first modified by it was first modified by Galileo and then by Robert Hooke. Okay, so this is the initial microscope development which was done by this scientist. Now, if we talk about Robert Hooke, he was one of the important known scientists in the field of this cell biology. And if we talk about Robert Hooke, he was a mathematician and physicist. He was a mathematician and physicist. And it is known that Robert Hooke developed a new microscope with which he studied the internal structure of number of plants. Okay, so what Robert Hooke did, he developed new microscope with which he studied the internal structure of with which he studied the internal structure of number of plants. His work is famous for the study of cork cells. Of cork cells. And Robert Hooke wrote a one book named Micrographia, in which he written all the detailed information about the some physiological descriptions of minute made by magnifying glasses with observation and inquiries thereupon. So they written a book named Micrographia. So this is what he did. Now in the cork study, he did a wonderful thing. What he did, he took a piece of cork from Spanish oak and prepared thin slices by means of sharp knife. Now then he used a lenses for throwing light on the cork piece, and then the piece of cork or thin slices of cork, these are observed under microscope. When they are observed under microscope, the piece of cork, the piece of cork was found to have was found to have a honeycomb structure. A honeycomb structure with numerous box-like box-like compartment. So this honeycomb structure was somewhat looking like this. The honeycomb structure was seen with the many compartments, and each compartment have a pore. Each compartment have a pore separated from other by wall. You can see here it is a pore, and this is the wall which is separating them. Like this structure he observed under the microscope. So later on, Robert Hooke Robert Hooke named the compartment named the compartment as cellulae. Singular is cellula, and now they are known as cell. So what do you mean by cellula or cell? According to the robot, it is nothing but a compartment with a pore and the wall which is separating them. So here the cell, if we talk, it is a Latin term. The cell, now it is known as cell. Actually, cellula is a Latin term. Cellula, it is a Latin term. And in this Latin term, the cella means hollow space or compartment. Now, Robert Hooke did not know about the significance of these structures and regarded them as passages for the conducting fluid. He initially regarded as a passages for the conducting. He does not know about the particular structure. Okay. Actually, the cell of Hooke were cell wall enclosing the spaces left by dead protoplast. It was initially the just dead protoplast which left the spaces, and that is surrounded by a wall. So the term cell, which was given by this particular Robert Hooke, actually a misnomer. Okay, actually the term cell, if we talk, is actually a misnomer. As a living cell is as living cell is neither hollow nor always covered by a wall. Nor always covered by a wall. Okay, so this is the what a particular Robert Hooke did. Now we know that prior to Hooke, the cell was also observed, and prior to Hooke, cell were also observed by Malpighi. And Malpighi, if we talk, in 1661, he called cell as sacules and utricles. And we have studied that Leeuwenhoek was the first to observe and describe scratch a free living cell. That also we have discussed. Leeuwenhoek was the first in 1673. He was first to observe, describe, and sketch a free living cell. It is known that he observed bacteria, protozoa, spermatozoa, RBC, etc. So that all he did. And in the beginning of the 19th century, it became clear that the bodies of organisms are made of one or more cells. And we know that the presence of nucleus discovered by Robert Brown. But you should remember that Robert Brown in 1831 discovered nucleus in orchid root cells. Okay, in orchid root cells it was discovered. Then it is known that Dujardin, now there is no need to remember this contribution of Dujardin, it is not getting asked, but I'm just for information, I'm giving you that Dujardin discovered living semi-fluid substance of cell and named sarcode and named it sarcode. Okay, Purkinje later on renamed sarcode which is given by Zardin as jelly-like substance, that is protoplasm. Then later on, cell membrane discovered by, we will discuss this in cell theory, cell membrane discovered by Swan in 1838, but name was given by Nageli and Kramer. And later on, slowly, as the electron microscopy has elaborated or discovered in a detail, the various organelles were also discovered inside the cell. Okay, so this is the history how different scientists made their contribution in the discovery of cell. But later on, the cell theory came into focus, which is the most important thing and which is one of the most important theory with respect to the discovery of cell. If we talk about cell theory, in this, in 1838, one scientist was there whose name is Matthias Sladen. He was a German botanist and he examined a large number of cells. Now, after the examination of large number of cells, like this animal and plant cells, mainly plant cells he observed, see, not animal cells. He observed that plants are composed of different kind of cells. What he observed? That plants are made up of different type of cells. And they observed that the cell is going to form a tissue. So he examined a large number of plant tissue later on, and he found that all plants were composed of made up of one or other kind of cell. Therefore, he concluded that the cell is the ultimate unit of what? Sladen concluded. Sladen concluded that cell is the ultimate unit of plant tissue. Okay. Now, who studied different type of animal cell? Then this was later when different type of animal cells was studied by Theodor Swan in 1838, who was a German zoologist. So Theodor Swan, 1838 he studied, and he was a German zoologist. He studied different animal cell or animal tissues, and he reported that in animal cell there is a thin outer layer which is known as plasma membrane in case of animal cell. And he found that animal cells lack a cell wall, and obviously they are instead covered by a membrane. So this is the cell membrane structure, which is look like this, which is covering the animal cell. Swan also concluded that presence of cell wall, it is a unique characteristic of plant cell, because later on he also studied the different plants and he concluded that the cell wall is the outermost covering in case of the plant cell. So later on, Swan proposed the hypothesis that bodies of animal and plants are composed of cell and product of cell. What they hypothesized? That either it is a body of plant or animal, it is composed of cell and its products. So later on, both combinedly formulated a cell theory, that is, Sladen and Swan combinedly formed a cell theory. Swan proposed the hypothesis that bodies of animal and plant are composed of and cell and product of cell. So cell is the basic unit of life. We know that. But later on, Rudolf Virchow observed that a new cells developed by a division of the pre-existing cells, that is omnicellula acellula, that is cells arise from the pre-existing cells by a division, then it is known as omnicellula acellula. So the new cells are formed from the pre-existing cells, and this was given by Rudolf Virchow. So initially Sladen and Swan proposed a cell theory, but it is known that the cell theory was modified by Rudolf Virchow, and that modified cell theory was further given. Now, what Rudolf Virchow did? He firstly explained that cells are divided and new cells are formed from the pre-existing cells, that is omnicellula acellula. It was not earlier to that. So, who modified the hypothesis of Slater and Swan? That is nothing but obviously Rudolf Virchow modified the hypothesis of Slater and Swan, and he added one more important statement that the cells arise from the pre-existing cell. Means the question may come that who given the final shape to the cell theory? Then obvious it is a Rudolf Virchow. So cell theory has a two components. All living organisms are composed of cell and product of cells, and all cells arise from the pre-existing cells. Okay, that is the important 2 points that has to be noted. And you should also remember that each cell is made up of a small mass of protoplasm containing a nucleus in it inside and a plasma membrane with or without a cell wall on its outside. And activities of organism are the sum total of activities and interaction of its constituent cells. Be remember, this is the third important point that you should not note down. Two components are there important, but third important point, this was added by Rudolf Virchow. Before that, one more statement was given by Sladen and so on, that activities of an organism are sum total of activities and interaction of its constituent cell. Okay, so this way the final theory was given. So this is the final shape to the what particularly, these are the actually having the three component. Okay, so actual cell theory was like this final cell theory. So we can say final shape to the to cell theory given by Rudolf Virchow. Okay, and this final shape given by Rudolf Virchow, it is known as, so this, so modified cell theory is also known as modern cell theory or cell doctrine or cell principle. Okay, that particular thing is ultimately concluded. So cell theory was credited to the work of, obviously it is Sladen and Swan who formulated cell theory. Cell theory cannot be applied to, yes, be remember this is one of the important point. Cell theory is not applied to the what some of the non-living things like viruses. Okay, it is not applied to virus because they are not neither living nor non-living. So obviously it is not applied to the viruses. Okay, some multinucleated structures, to that the cell theory is not applied. The largest isolated single cell is obviously it is an egg of an ostrich. We will discuss that in the further class. Longest cell, if we talk, it is a nerve cell. It is going to be the longest cell. If we talk about largest plant cell, acetabularia belong to acetabularia is a green algae. Okay, so it is going to be the algae, and we have discussed this about acetabularia and green algae in plant freedom chapter. Now we are going to discuss the overview of cell. That how cell look like what, the what is the shape of the cell and how it look like. So when we talk about the overview of cell, dear student, we come to know that which cell is typically a plant cell. Onion cell. Okay, if we talk about onion cell, it is a plant cell. And if we take a microscopic view of onion cell, it look like something this one. Okay, so here this is a particularly a whole boundary, and it is a space, and this is a nucleus. Okay, this is the nucleus, this is the space, and this is a boundary. So this is a cell. Now it is a distinct cell wall because it is a plant cell, obviously. So it is a distinct cell wall as it is the outer boundary, and just within it is a cell membrane. So two membranes are found. One is the cell wall, inner to that cell membrane will be seen in case of the plant cell. But when we see the human chick cell, it has an outer membrane as a delimiting structure of the cell. Outermost membrane is the cell membrane because cell wall is absent in case of the animal cell, and it is a human chick cell which is looking like this. And in this you will see the cell membrane is the outermost covering, cell wall is absent. Now inside the cell, in a dense membrane bound structure called a nucleus is found in case of the eukaryotic cell. Either it is a plant cell or animal cell, you will see the presence of a nucleus, a true nucleus, which is a membrane bound structure, and it is looking like this. Okay, in the eukaryotic cell. And in animal cell you can see, and in plant cell also you can see. In plant cell, okay, an animal cell, in animal cell this is the nucleus. Okay, and in plant cell also, if you see, this is the nucleus. Now what is the structure of this, we will discuss that when we will discuss nucleus in detail. Where are the chromosomes situated? Actually within this nucleus you will see that the chromatin material or DNA is condensed and present in the form of chromosome. So these chromosomes are present within the nucleus. When we talk about a chromosome, the chromosome contains a genetic material called DNA, and DNA is condensed in the form of chromosome. And DNA is somewhat like this, we can say the double helical structure, and which is condensed and form a chromosome, and chromosome are present within the nucleus. When we see the type of cell, there are two basic type of cells, prokaryotic cell and eukaryotic cell. As we have discussed now about the nucleus, true nucleus is found in eukaryotic cell but not in the prokaryotic cell. And these are some of the images of the prokaryotic and eukaryotic cell. If we discuss them in detail, then it is known that cell in eukaryote and prokaryote, if we talk, then in eukaryote, within a cell, you will see that membrane bound nucleus is present, but in the prokaryote, you will not see the membrane bound true nucleus. The lack of membrane bound nucleus is seen. And in place of nucleus, in place of true nucleus, nucleoid is present. Okay, now in both prokaryotic and eukaryotic cells, see, if you will see that semi-fluid matrix called cytoplasm occupy the volume of the cell, and maximum thing is occupied by the cytoplasm usually. So in cytoplasm, you will see it is a fluid-like structure, semi-fluid structure, which is present within the cell in both prokaryotic and eukaryotic cell. And cytoplasm, if we talk, it carry out majority of cellular activities. So it is considered as a main arena or main area. It is also called as arena of cellular activities in both plant and animal cell. Various chemical reactions occur in it to keep the cell in the living state. There are several reactions which take place in the, and chemical reaction occur in a, to keep the cell in a living state. So the question may be asked in exam that what is the main area of cellular activities? Then you should understand that is a cytoplasm. And this is the cytoplasm which is shown here in the cell. Then in eukaryotic cell, you will see the presence of membrane bound organelle, and some membrane bound organelle will be present like nucleus, vacuole, plastids, lysosomes. There's all membrane bound organelle you will see. You will see that nucleus has a two membrane, vacuole has a single membrane. In plastid, you will see the presence of two membrane. In lysosome, you will see the presence of single membrane, but membrane is present, which is having a structural similarity with the outermost plasma membrane, and it is found in these particular organelles. In mitochondria, you will also see the two membrane are present. In microbodies, you will see the presence of single membrane. In Golgi complex, you will see the presence of single membrane. And endoplasmic reticulum also you will see the presence of single membrane. So here the particularly membranes are present. Now how many membranes that we should understand. So it has a nucleus has a two membrane. So it is a double membrane bound organelle. Mitochondria has a two membrane. So it is a double membrane bound organelle. Vacuole has a one membrane. Microbodies has a one membrane. Plastid has a two membrane. So they are double membrane bound organelle, and Golgi complex has a single membrane. Lysosome also has a one membrane, and endoplasmic reticulum also has a one membrane. So this membrane or organelle which is found in the eukaryotic cell can be single membrane bound or a double membrane bound. So nucleus, you can see it is look like something this one. Golgi complex, it will be seen like this in a eukaryotic cell. Vacuole will be somewhat like this. You will see the vacuole. This one is the vacuole, which is occupy the large area in the cytoplasm of the cell, plant cell specifically. Plastids are also found. Plastids are going to be look like this. Okay, and these plasmids in this chloroplast are very well known, which carry out a photosynthesis. Then lysosomes are there. In lysosomes, if you see, it is single membrane bound structure. It is going to look like this. Okay, see lysosome. If we see here, it is labeled like this. Okay, this is a this is a particularly other organelles are there, and this is the lysosomes. Okay, now lysosome like this, a single membrane bound body. Then mitochondria, you will see that mitochondria is present within the cell, and it is, we know that powerhouse of cell which carry out a synthesis of energy. It is look like this, which is having a double membrane. Then endoplasmic reticulum look like this somewhat, which is associated with the nucleus. Okay, and it is of two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. Why they are known as like this, we will discuss in this later part. But when we talk about a prokaryotic cell, dear student, in prokaryotic cell, you will not see any such membrane bound organelle. Whereas non-membrane bound organelle are found in both prokaryote and eukaryote, and they are ribosome. Ribosome is found in prokaryote as well as eukaryote, but they are membraneless. So ribosome are there which carry out a protein synthesis, and they are made up of two subunits. We will discuss about ribosome also in detail. Now, apart from ribosome, centriole are also there, which are membraneless organelle, but it is found only in case of the animal cell. Ribosomes are found in chloroplast in plants, in mitochondria also they are found, and on rough endoplasmic or reticulum also you will see the presence of this ribosome. When we talk about animal cell contain another non-membrane bound organelle called centriole. And centriole, we know that, and we will discuss in the cell cycle cell division at how it helps in the cell division. So if somebody asks what is the main function of the centriole, you should answer that it is helping the cell division. And such a way cell get divided into the two. And you can see here that cell is undergoing division and it is divided into two. See, this is the how cell getting distributed into the two cells. How this occur, we will discuss in cell cycle cell division chapter. When we see the cell, when we observe a cell, you will see that cells differ. Different type of cells having a different peculiarities. That cells differ in size, cells may differ in shape, they can differ in activities as well. And when we talk about the size of the cell, you will see that mycoplasma is the bacteria, which is a wall-less bacteria, and it is the smallest cell. It has a zero point 0.3 micrometer in length. It is zero point 0.3 micrometer in length. And this mycoplasma, if we talk, they are also known as PPLO. Also known as PPLO, that is pleuropneumonia like organism. Okay, so such pleuropneumonia like organism are found, they are known as mycoplasma. If we see the bacteria, the size of bacteria is 3 to 5 micrometer generally, but the isolated largest single cell is found in the egg of ostrich, and the egg of ostrich, you will see, it is somewhat 3 to 5 centimeter long. It is, sorry, it is seen in the inches. Okay, so it is this much long, can be seen. Multicellular organism. Among multicellular organism, if you see, the red blood cells are there. They are about 7 micrometer long. Nerve cells, if we talk, they are some of the longest cells which are seen. Okay, these are the RBC. These are the nerve cells. Okay, so these sizes are seen in the. If we record a length or diameter of the other some important cells, then it is known that the RBC are obvious 7 micrometer in diameter, but some lymphocytes, if we talk, they are still smaller. They are 6 micrometer in diameter. Then, if we talk about cells of kidney, liver, skin, and intestine, and skin and intestine are 20 to 30 micrometer in diameter. Then, if we talk about muscle and nerve cell, we have discussed they are large sized. They are very large. They can be approximately 1 to 40 millimeter long, like striated muscle. And nerve cells are the longest cell. They are approximately 90 centimeter in length. They can reach up to this 90 centimeter length nerve cells. If we talk about among plant, the large cells occur in many algae. If we talk about internodal cells of Cara, they are 1 to 10 centimeter in length. And we have discussed that Acetabularia is a unicellular algae which is up to 10 centimeter in length. If we talk about plant fibers, that is phloem fibers, if we talk, they are 4 centimeter in cotton, 30 to 90 centimeter in jute, and over meter in hemp. In general, if we talk, we have discussed that eggs are large size because they store food for partial or complete development of embryo. If you talk about human egg, then it is also something slightly over than zero point 0.1 millimeter or 100 micrometer in diameter. So human egg, if we see, it is slightly over zero point 0.1 mm or 100 micrometer in diameter. We discussed that egg of ostrich is too large. So this is all about the different size of the particular cells. But when we talk about the shape, cells also vary in their shape. They can be round or biconcave. If we talk about the example of round and biconcave cell, then there can be red blood cells. Then, if we talk about another type of cell, they can be amoeboid type of shape can be there in a cell, like WBC. They are amoeboid in shape. If we talk about long and narrow cells, the columnar epithelial cells are long and narrow. Branched and long cells, if you see, they are nerve cells, they are longest cells. Tracheids cells in plant, they are elongated. And if we talk about round and oval, then mesophyll cells are round and oval. So you should remember this shape of different particular cells. Now, if we talk about amoeboid cells, they are looking like this. Long and narrow cells will look like this. The shape of the cell may vary with the function they perform. Yes, the shape also may vary with the function they perform. Elongated cells will look like this. They are elongated. Round and oval cells will look like this. Okay, so these are the different shape of the cell you can see in case of the different organism. Okay, now after this, let's discuss some of the MCQs. That animal cell is differentiated from the plant cell by the presence of, obviously animal cell is differentiated from the plant cell with the presence of centriole, and centriole is the single membrane bound organ, membraneless organelle which is found in case of the animal cell. If we talk about longest living cell is obvious nerve cell. Prokaryotic cell does not possess a nuclear membrane, obviously. Okay, because nucleoid is present in them. Okay, so this is all about the overview of the cell theory, and all introductory part of the cell chapter. Okay, I hope you understood about this. Thank you, students.